Good morning, everyone. Welcome to our webinar. First of all, thank you very much all for your participating in our webinar. We are seeing that several people are entering and starting the webinar. For whoever doesn't know me, my name is Luis Avila. I'll be hosting the webinar today. I am professor on weed science. On the Weed Science Research Group here in Pelotas. Pelotas is a city very south of Rio Grande do Sul, closer to the Uruguayan border. Here in our university, we have 6 weed scientists who perform research in a wide variety of weed science issues. In the last few years, we are dedicating most of our time for training the new generation of weed scientists, and this is the topic for this webinar series, to try to train the new generation of weed scientists with the new technology and finding on weed science. This webinar is promoted by the Weed Science Research Group. We are called Herb, is the Portuguese version of the Weed Science Research Group of our university, and by the Crop Protection Graduate Program, and is sponsored by the Brazilian Weed Science Society, which made available the webinar tool. This is financed by the Brazilian Weed Science Society. Before we start, I would like to mention a couple of information. During our presentation, you can ask question and answer system. We will gather the question, and at the end, Dr. Todd Gaines will answer the questions. Everyone who is registered in this webinar doesn't need to register to the next webinar. You will only going to receive an email with the invitation for the next webinar. Well, again, thank you all very much for attending our webinar. Let me introduce our speaker for today, Dr. Todd Gaines. Dr. Gaines is professor at Colorado State University and is a world expert on herbicide resistance. Today he will be speaking about novel herbicide resistant mechanism to synthetic auxin in weeds. Todd, thank you very much for accepting our invitation. It's a pleasure to have you here, and I will handle the screen to you so you can start your presentation. I will be here the whole time, so if you need any help, I'll be here.
Great, thanks Luis. Thanks for the introduction. Bom dia, good morning everyone. It's really a pleasure to have the chance to talk to you today about synthetic auxin resistance mechanisms. I think this is just a fascinating area about which we've learned so much more in the last few years. For those of you who were able to attend the webinar yesterday with Richard Napier from University of Warwick, you had a fantastic covering of how the synthetic auxin pathway works, how these herbicides function at the biochemical level in plants. And so today I'd like to build on that and talk about some additional resistance mechanisms that have been discovered recently. So again, thank you to Luis and the CHERB group and the Brazilian Weed Science Society for supporting this webinar. I just think it's wonderful that we can all still connect and talk to each other and learn from each other during this challenging time of COVID when we're not able to travel in our usual ways. So just a brief reminder about the synthetic auxins. Again, these were covered yesterday by Richard Napier, but the synthetic auxin herbicide group mimic the endogenous plant hormone indole-3 acetic acid, or auxin. And this is just an incredibly important hormone for controlling plant growth and development. It's a big part of the herbicide market for synthetic auxins that mimic auxin, and that includes things like dicamba, 2,4-D. As you can see in this fairly recent review, auxins compose about 9% of the world herbicide market. And there are, importantly, in addition to the traditional uses of synthetic auxins in grass crops, so particularly cereals, corn, and rangeland settings, turf, for control of broadleaf weeds, there are relatively recent traits that have been introduced, including the extend trait for dicamba resistance in soybean and cotton, and as well as the enlist trait for 2,4-D resistance in soybean and cotton that enable the selective use of these herbicides in those crops. So it's a very important mode of action group that continues to grow in importance with new discoveries as well, as we heard yesterday, particularly about Arylex and Rinskor. So when you look at the global use of synthetic auxins, again, a lot of treated area. 2,4-D globally is the highest at 162 million hectares in 2014, followed by dicamba with 50 million hectares. And then you can see there are a lot of additional uses for other auxin herbicides. These are often used in mixtures, in combination with other modes of action. So there's a lot of flexibility. And again, new discoveries such as haloxyphene methyl here, introduced recently from Corteva. So from the oldest herbicide 2,4-D to some of the most recent within the group. Now, again, a reminder, you saw this in more detail yesterday, but just in case you missed it or as a refresher, the primary mechanism through which synthetic auxins work for their toxicity is through interfering with this important process of plant growth and expression. So within the nucleus of the plant cell, there are a lot of genes that are called auxin responsive genes. So these include genes like NCED that lead to synthesis of the hormone ABA, ACC synthase that leads to the synthesis of ethylene, GH3, which is an enzyme that causes amino acid conjugation of auxins, as well as a type of genes called oxia genes. They're auxin responsive. And what these genes are is they're repressors. So you can see here in step number 1, this is an oxia protein cartoon, and this is sitting on top of a transcription factor that's called an auxin responsive element on the promoter of an auxin responsive gene. In this example, it's NCED. So you can see how this repressor is essentially blocking the expression of the auxin responsive gene. And this is the normal situation. Now, when you apply auxin, or when a synthetic auxin herbicide is sprayed on the plant, as in step number 2, that brings in this protein complex, part of which the receptor is called tier 1 AFB. There are several additional proteins in this I'm not showing for simplicity. But you can see that 2,4-D or the auxin will bind deep in a pocket in this protein, and that was shown with some really great illustrations yesterday, how this binding works. And then that's going to interact with a part of the oxia protein that's flexible to sort of dip down into that pocket and connect. And when this happens, that protein becomes ubiquitinated, which takes it away to be essentially destroyed. That frees up this auxin response factor promoter, the transcription factor on the promoter, causing expression of the gene, synthesizing the hormone or these other responsive factors. And in the case of a recent paper in Journal of Experimental Biology by Macaulay et al., they found that there's some suppression of photosynthesis related genes, probably as an effect of ABA blocking those. And through all these processes, collectively, the plant dies. But this is what's really critical. These repressors proteins are on top of the transcription factor protein repressing genes. And when the synthetic auxin comes in, it overloads the whole system. Now, I wanted to think of a way to illustrate this with a kind of an analogy. And so I have a 4-year-old son, and he's getting really into cartoons. And I had an idea for how a cartoon might kind of help illustrate this. So let's see how this explanation works. But again, we're thinking about the transcriptional repressor and the binding of tier 1 taking this away. So the cartoon I have in mind is The Incredibles, and particularly Mr. Incredible here. And in this analogy, Mr. Incredible represents the auxin responsive gene. So these are the things that make plants great, right? They're jumping up, growing through the regulation of ethylene, ABA, and other responses that are regulated by auxin. But you have these oxia repressors. And if you recall in the movie, when Mr. Incredible gets to the bad guy's evil lair, he runs into this trap, and you can see these sticky balls shoot out and start sticking to him. And even though he's very strong, they're just too much. And even Mr. Incredible can't continue to run when he's covered with all these black sticky traps that are shot out at him. And this is essentially, in a way, how these repressors are working. They actually form an aggregation over the transcription factor, and they're holding it down. So this regulation back and forth between, you know, the superhero jumping away or being held back by these repressors is really critical. And so that's why auxin is so tightly regulated normally in plant development. And synthetic auxins just throw this all out of whack. What they're doing, auxins or synthetic auxins, is they're taking away these sticky balls so that Mr. Incredible is free to run. And when you have too much of that, it's also not good for the plant. So when we're thinking about resistance, basically what resistance is doing is keeping these sticky balls on top of Mr. Incredible. So it's kind of like the bad guy synergy, and so that's resistance. That's keeping these repressors on top of those responsive genes. So you can let me know how you'd like this analogy. So when we think about in weeds synthetic auxin resistance mechanisms, it's quite amazing to think over the last several years the growth and understanding that we've had in this, because it really has been a mystery. These are herbicides that have been in use since the late 1940s, and resistance was first reported in the 50s to 2,4-D, but over time, relatively fewer cases of synthetic auxin resistance than others. So what do we know now about how this works? Well, with the understanding of this receptor co-receptor system, how do you slow down those auxin responsive genes, aka Mr. Incredible, to get resistance? Well, you could have more of those repressors. Just maybe one of those could overexpress to keep the responsive genes repressed. Maybe to have slower turnover due to less synthetic auxin binding target site mutations. There could be some transmembrane kinases that might interact with the auxin, and you could have less synthetic auxin arriving to this target site location in the nucleus. And so that could be due to changes in metabolism or translocation. And here's what we know to date. So this was an incredibly important discovery in kochia with the IA16 mutation of glycine to asparagine in the dagron. This paper is by Sherry Leclerc et al. In 2018 in PNAS. And Richard explained this yesterday, so I won't be going into more detail. But this was an indication that a mutation in the dagron of one of these AuxIA genes, IA16 specifically, can cause resistance to the herbicide dicamba. And then the two stories I want to tell you about today. So one is in Sesimbrium orientale in a different AuxIA gene called IA2, and there's a deletion near the dagron site. So we'll take a closer look at that. And the second story I want to tell you about today is a non-target site mechanism of enhanced metabolism. So this is happening in Amaranthus tuberculosis or water hemp, in which we see a 5' hydroxylation metabolism that inactivates 2,4-D, and this seems to be cytochrome P450 mediated. There are additional mechanisms out there that we know in a physiological sense what's happening, but we don't yet know at the molecular level. A really important one is transport. Yesterday, Richard was talking about the AUX1 auxin transport system, PIN proteins. So the movement of auxin and synthetic auxins from cell to cell is tightly regulated. And when you take a look at some weeds, in particular Raphanus raphanistrum or wild radish in Australia, you see at the whole plant level, there's reduced transport or reduced translocation of 2,4-D. This is speculated to potentially be due to change in ABCB transporters, so perhaps moving less 2,4-D around. There's also some indication of transmembrane kinases that may be involved, and these can potentially have some interaction with the AuxIA repressor proteins. So you can find more information on these in the papers by Danica Goggin et al. In 2018 and 2019. So very, very interesting story, and certainly more insights to come. And there are other examples of reduced translocation of synthetic auxins as well. And finally, an example that I think a lot of you in Brazil are familiar with is of non-target site resistance to 2,4-D is the rapid response in buva, in Canyza sumatrensis. So there's, when 2,4-D is applied within minutes and hours, you see a rapid cell death and leaf death. And so I'm really happy, I'm working with both Camila Pino's group at Rio de Janeiro And we have Jessica Leal and Amanda Souza, who are here at CSU right now, working on this. And Aldo Murado in Rio Grande do Sul is also working on this. You can see the publication in Weed Science just earlier this year describing this rapid cell death response. For some of these populations also have multiple resistance, five mode of action resistance. And so certainly we need to understand more about this rapid response. So I won't be talking about this in detail today, but I think collectively you can look at these 4 areas and see that there are a lot of options for synthetic auxin resistance. So that's certainly one take-home message I'd like to leave you with today. Now, next I'd like to talk specifically about the enhanced metabolism in water hemp. And this is one of the PhD projects for Marcelo Figueiredo here, you can see. So he completed his master's at ESALC with Pedro Cristofoletti, and he's been on CMPQ funding for his PhD here at CSU. And in fact, he'll be defending in just about a week or a bit over a week. So be looking, we'll send out on Twitter details on his PhD defense, and you'd be welcome to join and find out more details on what I'm going to share with you today. But 2,4-D is a great example because when we're thinking about detoxification and conjugation, the question comes up, and it came up in the webinar yesterday, why are synthetic auxins selective in grasses? And part of the explanation is likely due to the metabolic pathways. So when you're thinking about 2,4-D, we know that there's a UDP glucose conjugation that happens. So you can see here that 2,4-D is conjugated to this glucose sugar group. And also through GH3, one of those auxin responsive genes I mentioned, you can have conjugation with amino acids. This is primarily happening in dicot species. It does happen in grasses as well, but mostly in dicots or broadleaf species. And as I'll show you, these compounds have some reduced toxicity, but the interesting thing is that this reaction is reversible. So that means potentially these can be reactivated to the 2,4-D active form. But then what's primarily seen in grasses is P450, cytochrome P450 mediated reactions for various hydroxylations on 2,4-D. And these inactivate it, again, mostly seen in monocot grass species. And this reaction is probably not reversible, and then is followed by additional conjugations that allow this to be stored. So the faster this reaction can occur, can allow a plant to not have this synthetic auxin get to the target site and remove all those repressors and stimulate the uncontrolled gene expression. So you can kind of think of these are the 2 different pathways that are possible in plants. So when we look at water hemp or Amaranthus tuberculatus, a major problematic weed in corn and soybean in the U.S. and related, of course, to Palmer amaranth, Amaranthus palmeri. It's a diploid dioecious, so it has a lot of genetic diversity and produces prolific seed production. So a major issue. Also for multiple herbicide resistance, because it's outcrossing, you have populations that are resistant to 4, 5, 6 different modes of action. And you can see that it's quite widely distributed in the United States, but especially in the Midwest, upper Midwest, and Southeast Canada is particularly where it's a problem. So we're thinking about 2,4-D. The first report of resistance in water hemp was from Nebraska. Mark Bernards and Greg Kruger and colleagues are working on this, and their publication is in 2012 in Weed Science. But something you can see here, keep this in mind, these plants are incredibly resistant to 2,4-D. A typical use rate in crops is maybe between 200 to 400 grams per hectare, and depending crop situation. So you can see that use rate here, but going up to many-fold higher than that. Here's almost 9 kilograms of 2,4-D per hectare, and they're surviving that. They're not very happy, but they are able to survive it. And even here's 35 kilograms. A few plants are surviving. So it's a very robust level of resistance in water hemp. And Marcelo took these time course photographs. They're just really interesting, comparing susceptible on the top to resistant on the bottom. looking at, so each number is the number of days after 2,4-D treatment at 500 grams per hectare. So you can see susceptible, of course, within 1 day, you have the classic symptoms. The stem has bent over, leaf epinasty, and tissue death starting to happen, and this plant is never recovering. In the resistant, you do see these symptoms. Look, these leaves, they're drooping down. The stem is curling over, even getting some damage to older leaves, and the leaves are cupping. But at 4 and 5 days, look how this plant is recovering, and it stands back up, and by day 7, looks really not that damaged. So there's initial, you can see that 2,4-D is getting to that target site. It's activating some of these responsive genes, but then the plant is able to recover from that. And here's what Marcelo found. This is published in 2018 in Pest Management Science. This is the metabolic profile of 2,4-D in these plants. So let's take a look in the upper left corner here. This is susceptible water hemp at 264 hours after 2,4-D application. The 2,4-D has a C14 radioactive label on it so that we can measure it on an HPLC chromatograph. So you can see here on the x-axis, this is time in minutes, and the y-axis is number of counts. So you can see that we've got a metabolite here, labeled number 1, that separates from the parent compound 2,4-D is visible right here. So that is what's happening in susceptible and quite clear. But then in resistant, we have a different profile. You can see how there's less 2,4-D. So the parent compound of 2,4-D is much lower in number of counts. Metabolite number 1 is here, but there's not as much of it. There's more of what's identified as metabolite 2 and metabolite 3. And when we look at total composition of 2,4-D here on the right, in susceptible, you can see hours after treatment, how much 2,4-D is left, and compared to resistant, where the 2,4-D is disappearing much more rapidly. So then you can compare an I50, so basically number of hours for 50% of the 2,4-D to disappear. Well, that's 5 times faster in resistant than in susceptible. So Marcelo has gone further now to identify what are these metabolites. And metabolite 1 is an amino acid conjugation. So this is what would be predicted to be a conjugation by the enzyme GH3. And you can see that the conjugation is to an aspartic acid. And I'll show you, we have some phytotoxicity data on this metabolite. But what's happening in resistant is something different. So we find this 5' hydroxy 2,4-D. So you can see the 5' position on the ring is hydroxylated. This is a reaction that you'd predict would be performed by a cytochrome P450. And then metabolite 3 is actually a conjugation of that to a glucose. And then you go a little bit further, and that is malonylated. And so that then forms, that's metabolite 2. So you actually don't even see this 5' hydroxy in the chromatograph because this glucose conjugation and malonyl conjugation happen so fast. But we can identify it at lower levels, and it's there. Marcelo synthesized these metabolites. So you can see here just chemically how he's able to synthesize a 2,4-D aspartic acid. And then he was also able to synthesize this 5' hydroxy 2,4-D with also a chemical reaction. So he spent a lot of time doing these. And so I'm going to gloss over a whole lot of work just to show you the toxicology results. So he took 2,4-D and measured it on the model plant of Arabidopsis in a root growth assay. So you're looking at seedlings moved onto media containing these different metabolites at 7 days after exposure. So first of all, look at the black line with 2,4-D. So you can see this is relative root length where the dose of 0 is 100%. So you can see that at 0.01 and 0.1 micromolar, you're getting very strong inhibition of root growth, as you'd expect with 2,4-D. Then with the 2,4-D aspartic acid conjugate in blue, you can see there's actually sort of a growth stimulation effect at lower doses, but then you're starting to get 50% inhibition at about 1 micromolar. And so there's there's quite a bit of a reduction in phytotoxicity there. As you can see, that's about 100-fold reduction in toxicity. But then when you look at the 5-hydroxy-2,4-D, you can see it's having at 10 micromolar, no effect at all. 100 micromolar, just a little bit of effect. So the estimated dose for 75% inhibition is 232 micromolar, and that's nearly 20,000 times less toxic than 2,4-D. So we can say with confidence that the 5-hydroxy-2,4-D metabolite loses its activity as a synthetic auxin. Another really interesting experiment that Marcelo did is he took a system with a GUS reporter. So GUS, basically you get a blue stain in tissue that's expressing GUS. And this reporter system is activated by auxin signaling. So you can use it, it's used experimentally to measure whether a candidate receptor-co-receptor system is getting auxin. So he compared this with 2,4-D in these metabolites. So you look at this very low dose. This is what the Arabidopsis seedlings would look like in the stain normally. So there's like a little bit of blue, but not very much going on, right? Just at the tips. With 2,4-D at 0.1 and 0.5 micromolar, look at how much blue staining is happening around the tips and at higher doses. So this gives you an idea that's what 2,4-D is. It's certainly acting as an auxin. When you put the 2,4-D aspartic acid conjugate, you're really not seeing much until 1 and 10 micromolar. So similar to the phytotoxicity assay, but it shows that it does have some auxin signaling. And the 5-hydroxy-2,4-D, you can see at 10 and even 100, there's no signal. Only until you get to the very highest dose here of 1,000 micromolar, do you actually see a little bit of staining. So that's where it has auxin activity eventually, but you have to get to very, very high doses. Another observation that Marcelo had made earlier is that malathion synergizes 2,4-D in the resistant plants. So malathion is an insecticide, of course, but it's also known as a cytochrome P450 inhibitor in plants. And in the pest management science paper, you can see, again, here's a dose response with 2,4-D on resistant in red dots. But then when it's applied with malathion in this dashed line here, you can see it becomes nearly as sensitive as susceptible. So this is a further piece of evidence that cytochrome P450 is involved in detoxifying 2,4-D. And when he ran the chromatographs with 2,4-D treatment with malathion, that metabolite was lost. Then finally, this is data that's still at early stages, but we've been collaborating with Pat Tranel and Darcy Giacomini at University of Illinois to map this gene. And so the mapping data has produced some information that there could be something linked on chromosome 4 in water hemp. And you can see here there are 3 cytochrome P450 genes in that region. And Marcelo's conducted the gene expression measurements using real-time PCR. This one in particular, the P450 number 2, you can see that it's really not expressed in susceptible, but we do have expression in resistant. However, this is still very low expression, relatively. You compare especially to, these other genes are higher in resistant than susceptible to start with, but they're definitely induced by 2,4-D treatment. So this is where the work is going right now, is to understand whether one of these is actually the P450 gene that's responsible for metabolism, but that work is just still ongoing. So in conclusion, from Marcelo's work, you can see that metabolism of 2,4-D was 5 to 6 times faster in the resistant biotype. The susceptible plants do have metabolism. They conjugate 2,4-D to an aspartic acid. But the resistant plants perform this hydroxylation reaction that's normally only observed in grasses. Marcelo showed that these hydroxylated metabolites are less toxic than the aspartic acid conjugates. And the testing is ongoing for these candidate P450s for their function. Now, the second story I'd like to move to tell you about is Indian hedge mustard, or Sesembrium orientala. So this is a Brassicaceae weed, and this is a collaboration with Chris Preston at the University of Adelaide in South Australia. This mustard species is a problem in cereal grain production. It's a diploid that self-pollinates, also makes a lot of seed, very common characteristic for our difficult herbicide-resistant species. And it's resistant to 2,4-D, also to MCPA and to dicamba. So a bit more of a cross-resistance profile here. You can see this is another time course photo at 250 grams of 2,4-D per hectare. So a very different response here. Susceptible plants you can see at 1, 2, 3 days, they very quickly show epinasty, right? Look how the growth pattern is starting to twist. The leaves are spinning around, these leaves are cupping. So classic 2,4-D symptoms, and by 14 days, necrosis and the leaves are dead. Resistant plants show virtually no epinasty. So you can see that you get a little bit of maybe constricture on the leaf, a little bit of deformity, but basically it keeps on growing. And it's like the target site just is not perceiving 2,4-D. So Anita Cooper worked on this as part of her PhD before she finished, and she's now a lab scientist at Bayer Crop Science in Germany. But as part of her work, taking from Chris Preston, we had F2 seed, and so she self-pollinated those a couple generations to create recombinant inbred lines for a mapping project. And this worked out really well because we were able to have 6 recombinant inbred lines with a susceptible phenotype and 6 with a resistant phenotype. In Chris Preston's earlier work, they had shown that this was inherited as a single dominant gene. So we were able to have lines that recombine the genetic background from the 2 parents, but would only be different for the resistance allele. And we conducted RNA-seq on that. And without getting into the details of that too much, we assembled a de novo transcriptome. And between these 6 resistant and 6 susceptible recombinant inbred lines, it was amazing. There were only 3 genes that were significant based on a typical significance cutoff threshold. And one of them is a gene called IAA2. The other 2 that I'm showing here could have relation to auxin mode of action, but I would point out there was at least 1 recombinant inbred line that was inconsistent with that general pattern. So for example, this showed that resistant plants were generally overexpressing the ABCB13, but there'd be for example, a susceptible line that was overexpressing it, or a resistant line that wasn't overexpressing it. But this IA2 had a perfect association that there's lower expression of IA2 in resistant. So that was a little puzzling at first, until we took a look at the actual sequence of that gene. And we looked in a genome browser at the coverage, and what you're looking at here is we have 5 resistant lines compared to 5 susceptible lines. And there's this gap in coverage in all the resistant, and they all have it. There are no reads aligning right in this region, whereas all the susceptibles do have reads in that region. So I thought, could this be a deletion in the gene? And so we conducted sequencing and confirmed that yes, in fact, there's a 27 base pair deletion in IA2 in resistant. So that's shown here. This is in a very interesting spot as well. Yesterday, Richard was talking about the dagron of these oxi proteins, and particularly the Sherry Leclerc paper in which the glycine right here in this dagron region that's GWPPV was mutated to an asparagine, and that made it resistant. Well, in this case, the dagron sequence is still intact, but look right next to it, we have these 9 amino acids that are deleted. So this 27 base pair deletion keeps the protein in frame, so it doesn't disrupt the protein, but it's taking out a region that's called the dagron tail. And as I mentioned at the start, the oxi protein has to be flexible in order to kind of go into the binding pocket where auxin is in the tier 1 AFB protein. So this is a very interesting region. So we started looking into it, and we had a genetic association study, co-segregation analysis. So this is on a segregating progeny from a heterozygous parent. We had a Cas marker specific to the deletion allele shown in red and the susceptible allele shown in blue. And you can see over here, there's a perfect correlation. So if they have the susceptible allele, the intact allele, they're injured. If they're heterozygous, they show an intermediate injury. And if they're homozygous, they show almost no injury for the deletion. So that was a perfect correlation, great to see. And Marcelo took this a step further. He transformed the deletion allele as well as the wild type allele into Arabidopsis. So let's check this and see if it actually causes 2,4-D resistance. So here you can see Arabidopsis growing with an empty transformant, very happy, rosette, normal development. This plant is overexpressing wild type IAA2. No particularly remarkable phenotype, fairly normal. Here we have plants that are heterozygous for the IAA2 allele. So again, relatively normal development, normal rosette. But when they're homozygous for the IAA2 deletion allele, you can see they're very stunted, abnormal rosette, short. Something is really wrong with these plants when they're and keep in mind, this is under the 35S promoter, so they're really overexpressing it. And this is kind of the piece de resistance from Marcelo's work. So there's a lot to walk through here. What you're looking at is Arabidopsis plants grown on media and comparing the wild type to this deletion allele. So let's start over here in the bottom right-hand corner. This is Columbia 0, and you can see this is DMSO is the far right lane. So these are happy Arabidopsis growing normally. This is IAA, auxin at 10 micromolar. So you're seeing some root stunting, some abnormal development, and then 2,4-D and dicamba at effective doses for each of those. Half micromolar for 2,4-D and 5 micromolar for dicamba. Total inhibition of root growth, nothing happening there. We have lines just with an empty transformation. So they show the exact same response as Columbia 0. And then here you can see the two lines that are overexpressing the wild type IAA2. So it has the intact Dagron tail. Again, normal growth on DMSO, some inhibition with IA, and inhibition with 2,4-D and with dicamba. But then up here at the top right, here are two independent lines transformed with the IA2 deletion allele. First of all, you can see they have a phenotype just when they're grown on DMSO. They're potentially not growing roots as well, but relatively unaffected by IAA, relatively unaffected by 2,4-D. There is some root inhibition here, but they're growing a root, and same on dicamba. And then on the left, you can see this in the graph that under the dose of IAA tested, there's root inhibition kind of happening in everything. But when you look at 2,4-D, you can see that the two lines shown in red that have the deletion allele are showing much less root elongation inhibition compared to the wild type allele. So this is strong evidence that expressing the deletion allele gives a resistance to 2,4-D and dicamba. Now going further, we partnered with Richard Napier and Martin Kubes at the University of Warwick, and Richard was talking about the SPR affinity binding assay yesterday with tier 1. And so he gave the excellent explanation of how this is working. But basically, just to point out a couple of things in these data, this shows that there's a different binding association and dissociation for the deletion allele compared to the wild type allele. So this is done with a peptide of IA2. You can see the wild type contains the Dagron tail, and this part of PB1, whereas the deletion allele is missing this part of the Dagron tail. So the peptide is just this part and that part. And for all of these, when you compare, let's take a look at the binding of 2,4-D to wild type IA2 in tier 1. You can see there's a rapid association and a dissociation curve. But when you have the deletion allele shown in the blue dashed line, there's much less association happening, and it's also disassociating faster. Same trend for dicamba. You can see that there's more rapid association and slower dissociation for the wild type allele compared to the deletion allele. It's just not associating as well. And that what they're doing is they're fleshing in the synthetic auxin and fleshing it out, fleshing it in again and out. So you can kind of over and over check this. And when you calculate, you can see that the KD is much higher for the deletion allele compared to the wild type allele for IA, for 2,4-D, and for dicamba. So this gives us a mechanistic explanation. It doesn't bind as tightly when the Dagron tail is deleted, and it falls apart more rapidly. Now the question comes up of fitness cost. What about fitness cost? Because the IA16 mutation in the Dagron in kochia has been shown to have a pretty substantial fitness cost in terms of plant growth and seed production, particularly under competition. So we have data for this IA2 deletion, but not in competition. So you can see this is F5 recombinant inbred lines for resistant and susceptible, resistant shown in red and susceptible shown in blue. This is plant height over the growth course, and this is dry mass of the plants over the growth course. So really not much difference there, no difference. And seed production is very similar. So you can see that there's no difference in total seed production between resistant and susceptible inbred lines. So that basically makes us think, I think, the next thing is to look in competition, but at least in the absence of competition, when these plants are expressing the deletion IA2, normally they don't have much of a fitness cost for that. So kind of to summarize now, there are many paths to synthetic auxin resistance, and kind of coming back to the silly analogy, Mr. Incredible covered in these black sticky balls, he can't run away, and these are like the auxin repressors. And for resistance, you want to keep these on here because then you're not turning on all the auxin responsive genes when synthetic auxin gets applied. So how can that happen? Certainly target site resistance. And again, this is a new insight based on Sherry Leclerc's work with IA16. These auxin co-receptors are really important. We found this deletion of the degron tail of IA2. It appears that the degron is not as able to access 2,4-D in the tier 1 binding pocket. And no apparent fitness cost in ideal conditions, but certainly we do need to do more testing under competition and stress. Because it seems that IA2 is not expressed that much under normal conditions. It seems to be induced in response to stress. One point I want to make is that we did a full transcriptome analysis for SNPs, single nucleotide polymorphisms, using SAM tools in our data set. And we did not identify this deletion because it's not a single nucleotide polymorphism, it's a 27 nucleotide polymorphism. So I want to make that as a point. Keep that in mind. If you have a transcriptome data set, take a look in the genome browser at your candidate genes, see what's going on there, because, and use tools that look for more than single nucleotide polymorphisms. When it comes to non-target site resistance, we see enhanced 2,4-D metabolism in water hemp that's P450 mediated. I think very clear that the 5-hydroxy metabolite is no longer phytotoxic, but what is the P450 gene that's causing that? We need to find out, and does it have a fitness cost? That is still unknown. There are other mechanisms out there fascinating for reduced transport, especially the rapid response and BUVA. I think, whatever that turns out to be is also going to give us some incredibly new insights into how 2,4-D works and how plants can be resistant to 2,4-D. And I think as a final point, it's really important to know that we need weed genomes to study these gene families, because AuxIA, there are 24 of these genes, and they vary across plant species. Tier 1 receptors, there can be 4 or 5, 6 of them. Cytochrome P450s, hundreds. Transport genes, dozens of them. So that's going to be important for us to understand how weeds are adapting to synthetic auxin herbicides. There are additional remaining questions as well. For these target site resistance mutations, Richard Napier pointed out yesterday that probably these are more likely to give cross-resistance patterns across synthetic auxin families. And for this IA2 deletion, we can see that it gives resistance to 2,4-D and to dicamba. So that holds up there, and I think we have more to understand. It's definitely a complex target site. The co-receptor is one part. The tier 1 AFB receptor is a part. It seems likely that if a plant's tinkering with that, there might be a very substantial fitness cost, but remains to be seen. There's a lot of components of the ubiquitination machinery that could potentially be involved in resistance that we haven't seen yet, but we may. And there are many different AuxIA genes. So will we find that there are certain AuxIA genes that cause resistance across species, across different chemical families of synthetic auxins? Remains to be seen. Is IA2 the important one, IA16? I think very open questions there. Then when it comes to metabolism, we need to understand whether a given, in this case, a P450, does that give cross-resistance across synthetic auxin families or not? And what's the potential for cross-resistance to other modes of action? I think since the report of the population in Nebraska, there have been water hemp populations reported in Illinois and in Missouri. And these also have resistance to HPPD inhibitors. They have resistance to very long chain fatty acid elongase inhibitors like metolachlor. So what is going on with these complex metabolic cross-resistance patterns across mode of action? That is a really key question for the future. When it comes to transport, many candidate genes, a lot more to understand, and rapid response at this point, just a fascinating open question. And it's great that there are people working on it, and I think fairly soon we'll find some more answers to that. I wanted to end by mentioning the International Weed Genomics Consortium. So this is coming together in an international effort. A lot of you in Brazil are involved. If you aren't and you'd like to get involved, please reach out and get in touch. Because in particular, we have species like Cniza sumatrensis and Digitaria insularis on our list of species to do a complete genome for. And what we want to do is sequence these new genomes. We will have a website with analysis tools available so that you can learn how to access genomic data, analyze your own data sets. And then also we want to foster networking. So more collaboration, public and private, more coordination, more training, all of it driving new discoveries around weed management. So we're working in kind of the very late stages of formalizing agreements with BASF, Bayer, Corteva, and Syngenta. So we really want to thank them as well as the Global HRAC for their sponsorship. We'll have the first meeting of the, hopefully, the IWGC in April 2021. So look for more information on that. I also wanted to mention that Frank Dion and I here at CSU, because we're moving more and more instruction online, we're going to be offering an online class on herbicide mode of action starting August 24th. So it'll be offered through Colorado State University. And that'll be available for anyone to sign up for and take it as it's a 16-week class, and it'll go comprehensive mode of action, use of herbicides, and resistance. So if you're interested in that, please reach out to myself or to Frank Dion for more information, because we can work with anyone, anywhere in the world for that online class. We're really excited about it. So with that, I'd like to say thanks. Corteva in particular provided funding for this research, as well as CNPq for supporting Marcelo's PhD. I really want to thank Marcelo. He's defending his PhD next week and just has done fantastic work on this. As well, my colleagues here at CSU, Frank, Scott, and Phil, Anita Cooper did her PhD on this, Chris Preston, Adelaide, Greg Kruger, Richard Napier, and Martin Kubis with the collaboration on the SPR. It's been fantastic. And already Reddy here at CSU has helped us with their Rapidopsis work, and Pat Trannell and Darcy Giacomini for the mapping. So again, thank you for your attention, and look forward to the discussion and answering some questions.
Hey Todd, it was a great seminar. Thank you very much. It was very interesting how the deletion changed the response to the 2,4-D. I'm pretty sure that you are all very pleased to see the results, and it was interesting that still the machinery work even though all the deletion that happens. It's impressive. Yeah. We don't have any question on the board, so I have a question. You may share if you can, or if you can't, you don't need to share about the rapid necrosis. Do you have any hint what's going on there?
No, I think it can kind of speculate, and I've spent a lot of time with Camilla and with Aldo, understanding what they've done. But when you see that when 2,4-D is applied, it does have a pretty quick response in the sense of normally plant epinasty within several hours to a day, and a lot of these auxin responsive genes do get turned on within minutes. So that's there, but what you see in terms of this rapid cell death is more like a biotic response. For example, programmed cell death in response to a pathogen. And so that's kind of an intriguing hypothesis to pursue. Is somehow has the plant have pathways for defense response, been utilized for defense against the herbicide. And potentially those leaves where 2,4-D is applied die so quickly, and you can almost kind of see like the spray droplets where it hits, right? They die. And then is that, you know, that 2,4-D, it doesn't have a lot of translocation, but it does have some. And so is that kind of limiting how much is translocating to the growing point so that it doesn't get exposed to as much 2,4-D and it's able to hang on and recover? But then, you know, how is 2,4-D perceived to trigger these cell death pathways, if that's even what it is? There's all kinds of ways that plant cell death can be triggered. So that, I think a combination of, you know, physiology work, biochemistry, genetic mapping is going to be necessary to untangle that one.
Yeah, it's a very, apoptosis is very tricky for several species, and there is several process that link to the cell death, and that's going to be very nice to see in the future your results. We have to.
Yeah, we're excited, and yeah, with I mentioned Jessica and Amanda, who are here on their sandwich PhD from Rio de Janeiro. I think they'll, yeah, hopefully be generating some data sets, and we'll see where it goes. We've got a question here from Kaio at Oregon State. So I love the Mr. Incredible analogy. Thanks. I didn't know it was kind of funny, but it seems to fit. So you said maybe you mentioned it, but could you expand a bit more on the lack of fitness costs and resistant? The degradant seems crucial for plant development. Some other proteins compensating the slower kinetics. Well, here's what we think is going on for that is that so this is a protein. So there are multiple auxins that are involved in holding back these auxin responsive genes. And so there's overlap, there's compensation in those. IA2 itself has pretty low expression under normal conditions. And it seems to be more so induced in response to stress when you look at its profiling in different experiments in Arabidopsis. So it could be that it's, this is Marcelo's idea, that it's kind of a protective auxin. And so if it's only induced in response to stress, the fact that it kind of wouldn't get degraded as well under normal conditions maybe isn't so important. Maybe it's not involved in that much regulation under normal conditions, only in stress. So that's where I think, you know, we grew those plants in ideal conditions, you see no difference. Let's put them under some stress and see what happens, or you know, that will give us some more insight. So the first question, Juan Velasquez asked, the ester formulation may have some effect on resistance. Well, it could be. You know, we know certainly that between the ester and the amine formulations that we see different effects on, you know, antagonism in grasses. So those formulations act differently sometimes. It may be, but you know, I think that once it's should mostly be affecting how much is getting in and into the cell, and we're really we're not seeing differences in absorption of it seems like the same amount of 2,4-D is getting in, it's just metabolized differently. It should be the same 2,4-D acid once it's inside. From Aaron Hagers asking, several water hemp populations have very similar resistance profiles, in particular group 27, HBBDs, group 15, the very long chain fatty acid like metolachlor, group 4, auxins, as well as others. However, it remains unclear if this is due to selection from herbicides from each group, or if the selection was due to selection from only one group. In other words, are we seeing these profiles because of 60 plus years of atrazine use, 50 plus years of group 15 use, 70 plus years of 2,4-D use? Great question. And because I think in the case of 2,4-D resistance in that water hemp population, it was sort of a surprise, right? That wasn't necessarily expected. And then I guess my thought is that probably, especially when it comes to metabolic resistance, it may be that we've selected for kind of a master regulator of metabolism. So that these resistant plants kind of have a whole stress response pathway that's been selected. And there are lots of different genes like P450s, GSTs, glucosyl transferase, et cetera, that are all coordinated. And then when you have a genotype that all of these are getting overexpressed, maybe one of them works on 2,4-D, by chance, another one happens to work on an HBD inhibitor. By chance, that same network upregulates a GST that works on metolachlor, or maybe all that selection pressure with atrazine caused a general upregulation of GSTs that then also have activity on metolachlor. It seems like there's some linkage there that when you're selecting with a lot of different modes of actions, you're more likely to select for metabolic resistance. In fact, Paul Neve and his group at Rotham said they have a paper that's just come out with based on modeling work and work in blackgrass that when we often recommend mixtures of different modes of action for managing resistance. That's very effective for target site resistance, and it certainly delays target site resistance. But it may be that that mixture is more likely to select for metabolic resistance, because then you have kind of a general purpose, broad spectrum mechanism. So, sorry, Aaron, this is kind of some speculation, but I think that there's this is the question we need to answer, and there's more to do. From Frank Dion, we've got a question: how will resistance to auxin herbicides affect the future use of resistance traits? And so I think you mean especially probably in crops, transgenic crops, right? And you know, it's a big issue because there's such a cost to discover and develop those resistance traits over time that, you know, if you're looking at an older herbicide, in this case, they had the synthetic auxins that were out there for decades, and then put a resistance trait into crops where they hadn't been used. But a lot of the weed populations have already been exposed to selection pressure from, you know, other crops in rotation, like corn or wheat. And so I think that's a really tricky thing, to take an older herbicide, make a new resistance trait for it, and put it into a crop when you might already have quite a bit of resistance potential out there. Where in the sense of, you know, if someone discovers a new herbicide and they develop a resistance trait in the crop at the same time, maybe that has more potential, but it's also really difficult to do. So yeah, I think we've got a lot to learn and to see what's happening with auxin herbicide resistance. But then, you know, more traits will get stacked on top of that. Will the auxins still retain utility? Yeah, I think it's definitely an open question. And then, so I also see a question from Richard Napier here. Have you looked for resistance or its absence to other auxins in the IA2 plants, either in mustard or Arabidopsis? So yes, and this is where the answer is a little inconclusive, because we're doing these root growth assays, and for 2,4-D and dicamba, it's very clear, but we've also looked at fluroxapyr, picloram, clopyrrolid, haloxafen, and with the root growth assay, we're not quite so convinced by what we're seeing, and we want to look at the post-emergence treatment with those on the plant. But in general, they are resistant to several other of different synthetic auxins, but there are a couple that they're not resistant to. And off the top of my head, I'm not remembering, but there's not a complete cross resistance pattern, but there are additional chemical families to which we see cross resistance. All right.
All right, I think that's all the questions we have. All right, I think there is all the questions we had. Todd, thank you very much for your presentation. Was excellent. And I would like to thank the audience for the attention, and hope that everybody can visit us in the next webinar. Todd, thank you very much, and I hope to see you soon. Bye-bye.
Likewise, Luis. Thanks.
Bye-bye.
Okay, thanks everyone. Bye.